Hi everybody, DrBiology here, and I hope your exams, GCSEs, are going well. And paper 1 went well. I could see some of my predictions came up, so really pleased with that for you. So this is just a quick video about paper 2 revision. So I can't cover everything on paper 2, but I'm going to cover most things in this video, which will help you with your final revision for your exams. Okay, so paper 2 is going to be on Friday, 7th of June, and it's in the afternoon, PM, afternoon, and it's 1 hour, 45 minutes. Just a quick summary. Obviously, hopefully you've revised everything. You can check out some past papers, some topic-based exam questions on DrBiology. Learn the keywords as well, so there's some information there about learning the keywords, which I think you're probably pretty good at the moment. Know your command words: explain, describe, evaluate, calculate, suggest, things like that. Right, okay, so without further ado, let's get on. So the first topic is homeostasis and response, and there are some keywords here, so things like hormone, chemical messenger produced in glands and carried by the blood to specific organs in the body. Gland is an organ or tissue that makes a substance for release, such as a hormone. There you go, its target organ is the organ with a receptor molecule on its cell surface which recognizes a specific hormone, which is very important when we talk about insulin. Definitely know your hormones, particularly if you're doing foundation tier. They do like you to be able to label certain things, or at least know the parts of the different glands and what they do. Okay, so there's a good one, so I would probably pause and have a look at those. Menstrual cycle does come up regularly, so if you're going to be doing higher tier, you need to understand how different graphs, you need to check out different graphs of when hormones increase and decrease, but these are the main things you need to know. So you need to know the 4 main hormones, where they're produced and their role. So follicle stimulating hormone is made in the pituitary gland, causes an egg to mature, but also stimulates the ovary to release estrogen. Estrogen is made in the ovaries, so it stops FSH being produced, so it inhibits FSH production. It repairs, thickens and maintains the uterus lining, stimulates the pituitary gland to release LH. LH, luteinizing hormone, you just need to notice LH is made in the pituitary, and that triggers the ovulation. Now here's a little DrBiology top tip. If they give you a graph and they say when was the egg released, don't always go for day 14, because I'm pretty sure most teachers say day 14, which it is, but you need to check the graph, use the data in the graph. And then final hormone, progesterone, ovaries, maintains the lining of the uterus during the middle part of the menstrual cycle, and particularly during pregnancy. So, sense organs, another thing that they like, particularly with foundation students. Okay, so you need to know about the different senses, the receptors, and obviously the stimuli. So vision is the eyes and light, smell is your nose, which is chemicals, taste is your tongue, which is chemicals, touch, which is skin, and that is touch, pain, temperature and pressure, and then your hearing, your ears, both sound and movement. Reflex actions, this didn't come up last year, so you need to know what they are. They're fast automatic responses to stimuli. They bypass the conscious part of the brain. They may go into the brain, but not conscious part. They speed up a response by using what we call a relay neuron, and reflexes reduce your chance of being injured, so it's protective. So here's a diagram of a reflex action. So you can see that the stimuli is the flame, and obviously the receptor in your skin, and the way that it goes is through a sensory neuron and then across synapses, and we'll talk about synapses in a minute, through a relay neuron, so that's it can be in the brain, but this is in the spinal cord, and then back through the motor neuron to the effector. And remember, effectors are just muscles or glands. So I like to remember this by this this thing. So we go sensory, relay, motor. So we're going out, sensory, relay, motor. Got that. Synapses. Okay, so if you're doing foundation tier, you do need to know what a synapse is, but you don't need to know the full whack of how they work. So higher tier students, obviously it's an electrical impulse. Remember, it's not a message, it's an electrical impulse. Travels to the first part of the first axon. This triggers the nerve ending of a neuron to release the chemical messengers called neurotransmitters. And those neurotransmitters, they diffuse, so we get to 3. So they diffuse across the synaptic gap. And they bind with the receptor molecules in the number 4 on the membrane of the second neuron, and then the receptor molecules on the second neuron bind only to these specific neurotransmitters. Okay, and then it stimulates the second neuron to transmit the electrical impulse onwards. Reaction time might come up. Okay, so I'm going to put in the information bar down below, I'm going to put the link to my required practical video. Okay, looking much younger there. And then obviously investigating reaction times. So you might have done, well, you would have done this probably with different hands, your left hand or your right hand, or if you have maybe you've had some Coca-Cola or no Coca-Cola, those kinds of things. And basically your timing, the time it takes you to react, and usually to catching a ruler. Okay. What could we test? Well, there's lots of things you could test here. So I've just put what they might ask you about: taking drugs, gender, age, amount of practice. Remember, those are the independent variables, so the things that you are testing. What will you measure? Well, usually you drop a ruler, you record the distance in centimetres, and you compare it to a table. Usually they'll give you some data, and then you can calculate your reaction time. So this is the dependent variable, the factor you're going to measure. So in this experiment that I give you, it's the distance the ruler drops before it is caught. Control variables are, you'd use the same ruler, the same distance between the hand and the ruler. Can you see that? Yeah. So you keep the same distance. You don't touch the hand with the ruler. Use the same person dropping the ruler. Same position of the arm as well. Okay. So these are the factors you would keep the same. You could even say not having any stimulants before, not drinking any tea or coffee for 24 hours, things like that. Mean, median and mode. So that could come up. So in terms of mean is the average. They might ask you to calculate an average. The median is the middle of a sorted list of numbers, and the mode is the number that appears the most. So mean in this case is 11. The middle is 12. And the mode is 13. Resolution as well, remember, is the smallest measurement in an instrument. So the smaller the resolution, the more precise your data will be. Ways you could improve, yeah, I don't go on to that. The ways that you could improve this experiment is obviously you could use, for example, light gates, for example, so it would give you a more accurate result. You could repeat the experiment and find a mean, is really important. Okay, it also allows you to work out if there's any anomalous results that you need to remove from the calculation of the mean. The brain. Okay, brain didn't come up last year at all, so you need to know these 3 parts that are specified in the spec. Okay, so you've got the medulla, unconscious activities, heartbeat and breathing, the cerebellum, which is the coordination of muscular activity, the cerebral cortex, consciousness, intelligence, memory and language. Investigating the brain, so this could be a more applied question, and this would be higher tier only. So you might need to know, they might give you information about electrical stimulation, so you're stimulating a motor area of a particular part. The patient makes an involuntary movement. Also if a visual area is stimulated, they might see a flash of colour. Okay, so EEGs, you don't need to know what it stands for, can be created and studied to observe the electrical activity in the brain. More likely, I think, is about MRI brain scans, okay, because they use a strong magnetic field and radio waves to show details of brain structure. And it's got some MRIs do let the patient move. Okay, so there's some real advantages of that. So you can ask people to do different tasks. You can see which parts of the brain are active or not active. You can compare with a person who has or doesn't have brain damage, and you can actually see where the damage is as well. Because usually, traditionally, MRI scanners cannot be used if people can't stay still. So that's difficult if you're looking at a child or patients that have nervous coordination disorders such as Parkinson's. Okay, and also it helps if you're claustrophobic as well. Control of body temperature. This is biology only, and this did not come up. Okay, this could be a good 6 marker or a 4 marker, or at least 4 marks. So you need to understand what happens when your core body temperature is too low or too high. So in this case, too low, blood vessels supplying the skin, they constrict, so they get narrower, less blood flow to the skin, less energy heat is lost, shivering by the muscle contractions, releasing energy from respiration. Do not say that blood vessels move. They do not move. They either constrict, narrow, or dilate, which is get wider. All right, so just be careful with that one. Again, too high, so blood vessels supplying the skin, they dilate. More blood flow to the skin, more energy is lost to the environment, therefore cooling the blood. Sweat glands in the skin release more sweat. Sweat evaporates, transferring heat energy from the skin to the environment. Okay, so on to plant hormones. So auxins, they basically are mostly made in the tip of the growing stems and roots. These are known as meristems or apical meristems, and they diffuse to other parts of the stem. So if it's in a shoot, they usually diffuse downwards. Okay, so the 2 types of tropisms we're going to need to know about is phototropism, where things are sensitive to the light, and gravitropism, when sensitive to gravity. So all of this is to do with stems with phototropism. So either stems grow towards the light, which is positive, or negative, stems grow away from the light. Gravitropism, if it's a negative, it's about stems growing against the force of gravity, so that's going upwards, whereas roots grow in the direction of the force of gravity, downwards. Okay. Here's a quick summary of phototropism. So you can see when a shoot tip is exposed to light, more auxin accumulates on the shaded side of the root tip. The auxin's made in the root tip, remember, and it basically diffuses down in the shaded part. This makes the cells grow, or what we call elongate, so they elongate faster on the shaded side, thus bending towards the light. Negative gravitropism, okay, when a shoot is growing sideways, gravity produces an unequal distribution of auxin in the tip, with more auxin on the lower side. So this causes the lower side to grow faster, bending the shoot upwards. Okay. So other plant hormones. So if you do higher tier, there might be some smaller questions on gibberellins. They initiate seed germination. Ethene in bananas, important for ripening of fruit. And auxins can be used as things like weed colours, rooting hormone, or promoting growth in tissue culture. There is a required practical here, okay, so you need to make sure you go through that required practical. I again have put that in the box down below so you can have a look at that. Right, so we've done homeostasis and response, so it's quite a lot actually. So inheritance, variation and evolution, definitely know your keywords. I'm not going to go through them here, but I'd like you to pause and know your keywords. A key thing that might come up, because it's quite important, is the differences between asexual and sexual reproduction. So in terms of knowing, comparing or contrasting or evaluating. So you can see there's advantages of asexual and disadvantages of sexual. Again, I'll go through that. DNA structure, basic DNA structure. It's a polymer made of 4 different nucleotides repeated in different sequences. So you need to know that thymine pairs with adenine. Okay, so I say things like the tree and the apple, so thymine and adenine, and guanine pairs with cytosine, a car goes in a garage. That's how I remember it. Okay. Genetic disorders, it possibly will come up that genetic disorders, but don't expect just the 2 that you've learnt about. There could be many. Okay, again, you need to know keywords. So you need to know what dominant recessive alleles are, homo and heterozygous. So here's a disorder by a dominant allele. Okay, they could show you a different disease. So be very, very aware of what type of allele determines the genetic disorder. So remember, it's a mutation. It's a mutation of an allele. And polydactyly is when you have extra fingers or toes. It's caused by a dominant allele, so it can be passed by just one allele. So here we've got parents. They usually give you the parents' alleles, and you can see that the father has it and the mother is unaffected. And they'd expect you to do a Punnett square, and I suggest you do Punnett squares. I only teach my students Punnett squares because I think it's just easier to do. So if we look in this example, okay, you can see that the student here has put in the alleles, but also said which ones have it and which ones don't have it. And sometimes they'll want you to work out a probability. Okay, so in this case, 50% of offspring, there's a 50% chance of getting polydactyly. Recessive alleles, so cystic fibrosis, they could show a different one again. I always teach my students thick, sticky mucus, basically. That's the symptoms, and it clogs up the lungs, digestive system, causes infertility as well. And basically, you've got to inherit 2 copies of the recessive gene. Okay, so in this case, the parents, one of them, basically, they have one allele that is causing cystic fibrosis. So these parents are called carriers. So they don't have the symptoms, but they do carry the mutated allele. So again, here we go. So we cross it, and then you can see that actually, in this case, there's a 25% chance of having cystic fibrosis. There's a 50% chance of being a carrier, and there's a 25% chance of not having anything related to cystic fibrosis. So there we go. Genetic modifications, genetic engineering, again, they could maybe give you some data or some information. You do need to know the method, so they cut out a gene from one organism using enzymes. You don't need to name the enzyme. And then the gene is added to other organisms, DNA or chromosomes, and then the gene is added to an early stage of development. They might ask you about insulin production. So insulin uses bacteria. So you produce, you put the human insulin gene into a plasmid in the bacteria, and then obviously, because they asexually reproduce, they clone and they can produce lots and lots of copies of the human gene that releases insulin. Evaluating GM crops, I'm not going to go through the benefits and risks. These are some examples that I've just taken out for you, and I think it's really important when you're evaluating. Usually they'll give you lots of information. When they do, they'll say use the information and your own knowledge. So here's some really good arguments for and against GM crops. Classification, I'm hopeful this will come up because I do love my mnemonic here. So the way to remember Linnaeus' system of classification, I use king prawn curry or fat greasy sausage. it does work. So that tells you the list. Remember, the binomial system is where it allows scientists to accurately identify individual species. So, for example, genus and species. So Homo sapiens, for example. Linnaeus didn't look at other things. He only looked at major animals and plants. So he didn't really look at fungi, moss, lichens, bacteria. He couldn't distinguish between them because they didn't have the technology to look at that. And these days we've got DNA advances, biochemistry, better microscopes for better kind of classification. Three-domain system, you will need to know the Carl Woese three-domain system. So archaea, bacteria, eukaryota. They also like you to be able to read evolutionary trees as well. Okay, ecology. Again, ecology, lots of keywords. Learn your keywords very, very carefully. So I would pause or maybe use your notes or revision guide and check you know your keywords. Carbon cycle, possible 6-marker this year, maybe. You need to know all the main things. Okay, so what I've done is I've taken an exam question and got the mark scheme, and this is describe how living things are involved in the constant cycling of carbon. And you can see the answer is there, and you can have a look at that in a minute. Key thing though is just remember that it's about cycling. So photosynthesis is going to remove carbon from the atmosphere, or carbon dioxide from the atmosphere. Okay, respiration is going to release it into the atmosphere. Okay, when things die, they're going to then be decayed and broken down by microbes that respire, so they release CO2. Carbon compounds, some things get buried, and that then they become fossil fuels. And the only way that carbon can be released from that is through burning and combustion. So those are the main kind of processes that are going on. Okay, so there's an answer to that question. You can decide to go for it or not. I don't know whether there'll be a 6-marker, but it hasn't come up for a while. Abiotic and biotic, again, expect data and lots of information. Remember, abiotic is the non-living factors. Biotic is the living factors. So the living things, particularly competition. They might talk a lot about competition. Adaptations they might talk about as well. Say what you see. It's common sense adaptations. Quadrats. Okay, so yet another required practical. So this one, there was a question last year, but they do like the answer. They do like asking questions about the use of quadrats, particularly either working out populations of plants on a field or using a transect. So that's a tape measure across different areas, like if you're looking at sand dune succession, for example, or different areas, and you're going to use your quadrat. So again, check out my videos on that. Trophic levels, you need, obviously, again, you need to know what all these things are. I'm pretty sure most of you know what those things are. And what you're going to use it for, well, I think you're going to use it for a pyramid of biomass. The sparrowhawk, it's pecking my head. So yeah, pyramids of biomass. Remember, if you're going to draw it, because they might ask you to draw it, so take a ruler, you need to check that the bars are equally spaced around a midpoint. You can't see the midpoint on there, but they need to be equally spaced, one side and the other. Width is always the same. Bars are touching. Producer bar is always at the bottom. So producer is always a plant. Could be a microbe, but it's not a microbe. It could be a fungi or something like that, but it's usually a plant. Length of each bar is proportional to the amount of biomass available. Okay, and you might have to calculate percentage energy transfer. So remember, it's the biomass in the higher trophic level divided the biomass in the lower trophic level. Okay, so here's just an example. Times it by 100 to get percentage. Decay, decomposers, fungi, bacteria, detritivores, worms, flies and maggots. Decay is controlled by temperature, water and oxygen. just need to remember those three things. Again, required practical, last required practical, there's lots in this paper, is the decay required practical. I'm looking quite scary there. I don't know what I was thinking about there. So I'm using an indicator called phenolphthalein. So again, worth checking out the video. Biodiversity. So you do know that human populations have grown, so that's going to cause issues for different habitats, okay, and biodiversity. Eutrophication has not come up for a while, okay, so eutrophication, maybe learn the different steps of eutrophication. Food security, that hasn't come up for a while, and particularly relates to climate change. I haven't mentioned climate change here, but they could ask you about climate change. And sustainable fisheries. Yeah, that hasn't come up for a while. And about how you conserve fish stocks. So you need to regulate net sizes. So you have large holes in the nets to let young fish escape. You impose fishing quotas. So fishermen can only take out a certain weight of fish. You limit fishing so that you don't allow them to fish during breeding seasons. You don't discard fish, which they used to, okay? And then you ban certain fishing in certain areas. So you have protected areas, conservation areas. Good. Okay, there's a lot there. So you might have to whiz through some of that yourself. Again, remember DrBiology. Here's my front page. And it might help you, and you can actually find the PowerPoint for this on the front page of DrBiology. Okay, hope you found that really useful. I really hope things go well for you in paper 2. Keep going, you're getting there, guys. You're getting through it, and that's the main thing. And never give up. Attempt every question. Okay, good luck. I'll see you soon.